Well, there were four big developments in biotechnology. It happened round about 1970 or slightly afterwards, and these really transformed the field of hematological malignancy diagnosis. And the first, which sounds a little bit arcane, was the ability to use bacterial enzymes to cut DNA into defined size pieces. And the reason why that was important is that following on from the studies of the germinal center, it was realized that lymphocytes rearranged the genome, and so that you could have a very accurate fingerprint of an individual lymphocytes based on the size of these DNA fragments. And that then allowed a much more accurate means of identifying monoclonality within something which you suspected as being a tumor. And that was called southern blotting. Now, the other three developments, southern blotting is now something which is basically obsolete, but there are three other things. The first is the ability to sequence DNA, which was the so-called Sanger method of sequencing DNA. And the second was the polymerase chain reaction, which is effectively a method of photocopying very defined segments of DNA. And the third was the ability to produce monoclonal antibodies. And all of these things have had a massive impact in hematology. It's actually one of the interesting things about hematology is that these things were adopted much more quickly into routine clinical practice than in other areas of oncology. Our late colleague here, Brian Roberts, who was a president of the BSH, was very much an enthusiast for this and drove locally here the introduction of these techniques into laboratory practice. The polymerase chain reaction has got numerous applications, but took over as a definitive method of identifying monoclonality, but also in picking up other mutations and genetic changes in cells, which became a central part in the diagnosis of different types of leukemia and lymphoma. And monoclonal antibodies, prior to the development of monoclonal antibodies, the antibodies that were used in immunocytochemistry were produced in rabbits and goats and sheep. And when the animal died, then that was the end of that reagent. Monoclonal antibodies can go on forever, and so it produced a highly quality assured, reproducible reagent that could be produced in almost indefinite quantities, used across the world, and that really transformed the whole process by allowing cells to be accurately typed and characterized as to what proteins they're expressing. And finally, the ability to sequence DNA eventually led to the Human Genome Project and the full sequence of the human genome, which in the later parts of the story play a key role in understanding hematological malignancies and in everyday diagnosis.